Good day, I'm Archelina Pierre taking a Bachelor of Science in Agriculture. In this video, I'm going to tackle about the structure of the DNA and the step-by-step of DNA replication. Structure of the DNA. DNA is made up of molecules called nucleotides. Nucleotides are connected together to make two long strands that winding to form a structure called a double helix. In the event that you think of double helix structure as a step, the phosphate and sugar particles would be the sides, whereas the bases would be the rungs. Step 1. Replication fork formation. Before DNA can be replicated, the double-stranded molecule must be unzipped into two single strands. DNA has four bases called adenine, thymine, cytosine, and guanine that form pairs between the two strands. Adenine only pairs with thymine and cytosine only binds with guanine. In order to unwind DNA, these interactions between base pairs must be broken. This is performed by an enzyme known as DNA helicase. DNA helicase disrupts the hydrogen bonding between base pairs to separate the strands into a Y shape known as the replication fork. This area will be the template for replication to begin.
This is a sample video of DNAization.
Step 2. Primer binding. The leading strand is the simplest to replicate. Once the DNA strands have been separated, a short piece of RNA called a primer binds to the third end of the strand. The primer always binds as the starting point for replication. Primers are generated by the enzyme DNA primase. Step 3. Elongation. Enzymes known as DNA polymerases are responsible for creating the new strand by a process called elongation. There are 5 different known types of DNA polymerases in bacteria and human cells. In bacteria such as E. coli, polymerase III is the main replication enzyme, while polymerase first, second, fourth, and fifth are responsible for error checking and repair. DNA polymerase III binds to the strand at the site of the primer and begins adding new base pairs complementary to the strand during replication.
This is the sample video of elongation.
Step 4. Termination. Once both the continuous and discontinuous strands are formed, an enzyme called exonuclease removes all RNA primers from the original strands. These primers are then replaced with appropriate bases. Another exonuclease proceeds the newly formed DNA to check, remove, and replace any errors. Another enzyme called DNA ligase joins Okazaki fragments together, forming a single unified strand. The ends of the linear DNA present a problem as DNA polymerase can only add nucleotides in the fifth to third direction. The ends of the parent strands consist of repeated DNA sequences called telomeres. Telomeres act as protective caps at the end of chromosomes to prevent nearby chromosomes from fusing.
Sample video of termination.